As of recording this video, it was yesterday, in fact, when I was browsing YouTube and I came across a video that was talking about the mechanisms of ricin and how it actually works as a poison on the body. Now, ricin, rather than being a chemical like most drugs and most poisons, is actually a protein, and the proteins are much bigger when it comes to a biomolecular scale than it comes to a chemical such as, I don't know, acetyl-CoA or citrate or something like that. A protein is much vast than something like acetyl-CoA, which is just made of molecules. And protein is made of hundreds and hundreds of amino acids, and I'm pretty sure ricin, if I can remember the exact figure, half of the protein, half of the ricin protein, ricin toxin B, I'm pretty sure it is made up of 529 amino acids, which is an insane amount. And then you sort of combine that and multiply that by 2, so you get ricin toxin A and ricin toxin B. That's going to be around over 1,000 amino acids, and amino acids contain like bare minimum 7 atoms per molecule, so that is going to be a lot of atoms. But when this video actually got to how ricin affected the body rather than its structure and all that sort of stuff, there was a very ear-catching figure which got me thinking and sort of had a lot of thoughts that trailed off of it. It was something to do with how the ricin toxin A, which is half of the protein, ricin toxin A alone is not able to actually get into cells, so it can't actually have its effects and mechanisms on the body. But when it is ricin toxin A and ricin toxin B lets it go into cells. Then the ricin toxin A and ricin toxin B is going to be split by certain like digestive enzymes. Then ricin toxin A, which is the bit that actually does the stuff to the cell, then it's going to go ham on all the ribosomes inside of the cell. It's going to cleave an adenine off of the sort of stabilizing bit of the ribosome. It's going to clean this adenine molecule, which is used to stabilize the amino acids, which is used to make chains of proteins, such as digestive enzymes, such as just enzymes that are needed to make things into energy and all that sort of stuff. So now when this adenine is cleaved off, the ribosome cannot work properly, and this means that no proteins are made from that ribosome. And while you may think, oh, that's just one ribosome, there's going to be billions of ribosomes in our body at one point in time. And this ricin toxin A is probably going to only cleave off maybe 1 per second, or maybe 1 per every couple seconds or something like that. But this was where the ear-catching figure really came in. It cleaved off, and like one ricin toxin A molecule cleaved off an adenine off of a ribosome at a rate of something so heinous per minute, it genuinely scared me. It was something like 4,000 ribosomes dead and useless per minute, which is absolutely insane to me. And that spiraled off a lot of thoughts that how quickly does all this stuff, the glycolysis, the Krebs cycles, actually happen inside of our body? And the answer to that is undefined. As much as I tried to research it, it was an incredibly hard sort of thing to find, because these sort of processes, glycolysis, they have enzymes. They have enzymes that take glucose all the way to pyruvate, and then pyruvate will go through the Krebs cycle into acetyl-CoA, through the Krebs cycle, oxidative phosphorylation, all that sort of stuff. All of these enzymes may not necessarily be available at a certain time, so you're going to have a molecule of, I don't know, glyceraldehyde that's just waiting to be turned to the next stage in glycolysis, but there's just no enzymes available for like the next 4 seconds. And even though one singular enzymatic reaction, it takes a millisecond, that's not going to mean that glycolysis, which has maybe 12 steps, takes like 12 milliseconds, because that will be incredibly quick and also incredibly hard to imagine. So one process of glycolysis, as an estimate and as backed up by some of the things that I found during my research, takes around maybe 4 seconds as a general average. That is like the anchor, because there's not going to be a continuous line of enzymes. There's not going to be perfectly available at any given time. It's going to take an enzyme when it realizes that it needs to be somewhere, and it's signaled by another part of the cell to get there. And while enzymes are going to be quite abundant inside of cells, it's going to still take quite a long time, right? At least in terms of how like these processes relatively work. So maybe 4 seconds, because there'll be like slight breaks between all the steps. Even though the enzymatic reactions themselves happen quite quickly, it's going to be a little bit staggered. So 4 seconds, which is still incredibly quick, is probably a good base marker and an estimate. But there are a lot of chemical reactions that happen in the body, and a lot of chemical processes that are regulated. The production and biosynthesis of cholesterol, which often happens at night, which I've discovered during my research, is regulated by the enzyme HMG-CoA reductase, because cholesterol, the product itself, regulates how much of HMG-CoA reductase can be made. So it's sort of a self-regulating process, which means if which means that the whole sort of rate of reaction that can occur to get from acetyl-CoA to cholesterol takes quite a long time. And while if everything happened quite smoothly, if everything was available at the same time and it went straight from all the acetyl-CoAs that are used and all the other products that are used all the way to cholesterol, if HMG-CoA reductase was available at that particular point in time, I personally think it's probably going to take around a minute, and that still is incredibly quick for one cholesterol molecule to be produced from acetyl-CoA, going through all the chemical, and that's going to be happening around, I don't know, 100,000 times in every single cell of your body at any given time during the night when it's most commonly made. I think it's during noon, around 9 o'clock in the morning to 12 o'clock noon, where it's made the least and the rate is slowest, which is quite an interesting thing, because I guess cholesterol is needed for other stuff during that time. And so, I don't know, your body and your biochemical processes are an incredibly complicated thing. Another thing that I've also realised is that the rate of these chemical reactions can also be sort of skewed if one was to try to experiment them. Because if you think of glycolysis, again, you've got glucose to pyruvate. There are certain molecules inside of the process where molecules can be fed into this process. For instance, the metabolism of fructose. Fructose can be metabolized into G3P, which is a product of glycolysis sort of midway through the process itself. So you can't really tell if that G3P's been from glucose or been from fructose. It's sort of a weird thing, and it's quite hard to understand that. So it's incredibly hard to find an estimate, and as much as someone who's a bit compulsive and obsessive over these sort of interesting things like I am, it was very, very hard for me to find, and it took like an hour of research just to really get nowhere, and every source telling me that it's just not something that can be defined and quantitatively set. But with that, I recommend subscribing, because I like this community of young tribal people who are willing to improve themselves so much more than we already have, and good luck.